So good morning, everyone. We're going to start the session. It's my pleasure. So I'm Catherine Dubourdieu from Helmholtz-Zentrum Berlin, Germany. And it's my pleasure to welcome Dr. Rama Vasudevan for this plenary talk. Dr. Vasudevan is a senior R&D staff scientist at the Center for Nanophase Materials Sciences, CNMS, at Oak Ridge National Laboratory, and is the group leader of the data NanoAnalytics group. He received his PhD in material science in 2013 from the University of New South Wales with a thesis on scanning probe microscopy of electric materials. He joined then the CNMS on the same year as a postdoctoral researcher and became a staff scientist in 2016 and was promoted to a group leader in 2022. Together with colleagues at CNMS, he won the Microscopy Today Innovation Award in 2019 for developing software to automatically extract atomic coordinates of atoms and defects from noisy microscopy images. Also with colleagues at CNMS, he won the same award again in 2024 for developing a combined software-hardware device for automating microscopy systems with a universal interface. He was also part of a team that won an R&D 100 award in 2023 for an autonomous microscopy physics discovery platform. He is a fellow of the American Physical Society and has published more than 150 peer-reviewed papers, as well as being an associate editor of the IOP journal Machine Learning, Science and Technology. His current research interests include for electrics, scanning probe microscopy, and development of machine learning methods for better understanding and manipulating materials at the atomic and mesoscopic scale. So it's our pleasure to have you here today, Rama, and we're looking forward to listening to your talk.
All right, I hope you can all hear me well. Thank you, Catherine, so much for that kind introduction. Thank you to the Mexican Materials Research Society and the organizers here. It's a fantastic conference. I've been coming here for a number of years now. I've been involved in symposia organization. I've given invited talks. And so it's really my pleasure to be here and deliver this plenary and give you some insight into what we are doing at the CNMS with respect to autonomous experiments and where it can really take the field of material science into the future. Before I start, I'd like to thank all of the members of my group, and you can see their photos on this particular slide. The DNA group, the Data NanoAnalytics group, is a group at the CNMS, and we're all scientists by training. That is to say, we are either material scientists, physicists, or chemists, and we have backgrounds in specific areas of physics or material science, but we supplement that with an understanding of machine learning. And right now we are focused on automating scientific characterization and synthesis tools and really imbuing them with autonomous capabilities. And I'll explain why that's important and what we can get out of that, and why that's, I think, the future of the way that we're going to do science in the next couple of decades. So some acknowledgements first. This work was funded by the Department of Energy in the United States. And so I do work at a user facility, the Center for Nanophase Materials Sciences, which is this building on the right-hand side. There's a larger building on the left-hand side you see. That's the spallation neutron source. So if you've ever wanted to do neutron scattering experiments, you can write proposals to come to that center as well and perform a variety of different kinds of neutron experiments. But we are a sister facility to that. And so we're a smaller user facility where we help users to perform their experiments, and we also perform in-house research, and so that's part of my job as well. I'd like to thank all the collaborators and all of the users that have been involved, at the CNMS in the last decade that I've been there, and in particular for the people that have been instrumental in much of this work. On the right-hand side, you see Kevin Roca-Priori, who did a lot of the scanning transmission electron microscopy, Yong-Tao Lu, who's leading our efforts in atomic force microscopy, Sumner Harris, who's been doing pulse laser deposition and automation on that aspect, and then Stephen Jesse, who's my longtime colleague, who has sort of been a pioneer in automation and machine learning applied to microscopy. And so with that said, let's talk about materials challenges. So we know that for renewable energy, for things like sensors, for things like self-driving cars, for different kinds of displays, for low-power electronics, we need to discover newer and better materials, right? And so the question is, how are we going to discover these new materials? Traditionally, we would use serendipity, so we would use the researcher's intuition as well as some luck and hope that we can discover new materials. But things have changed, and in the last decade, we have found that we can actually predict new kinds of material compounds based on the requirements that we put on them. And so it is relatively, not necessarily straightforward, but it's certainly possible to be able to predict thousands of new kinds of material structures every day. And we can do this in a fully autonomous fashion using high-throughput computational approaches, right? Now, the challenge, though, is that the materials are very complicated these days. They're very complex, and they have very complex underlying physics. And we need to be able to understand that physics if we're going to be able to optimize them. And we also need to be able to synthesize these predicted compounds, right? And so this is where I think autonomous material science can actually really make significant inroads, right? If we can solve these two challenges of can we realize the materials that we are predicting and can we better understand them, then I think we're going to be able to tackle many of the material science challenges of today. Now, machine learning in materials actually goes back a very long way. And so you can probably go back at least to the 1990s, where there was a lot of work on combinatorial studies and combinatorial synthesis. And so high-throughput combinatorial studies gave us significant amounts of data. And so when people were looking for how they could process that data, then they turned to machine learning approaches. So it's not actually very new, but the thing that happens is that whenever there's interest in AI and machine learning, there is a concomitant increase in interest from other domains, and then when that interest wanes, for example, because of AI winters, then at the same time, the interest from other domains will also wane. And so there's sort of these peaks and troughs in terms of interest of machine learning within the material science story. And so if you go back, for instance, in 2001, there was an MRS symposium entitled Combinatorial and Artificial Intelligence Methods in Material Science. So that's, almost 24 years ago now. If you read the abstracts in that particular symposium, you will find that the abstracts are very similar to the abstracts that are being submitted today, right? And so what this is basically saying is that we have been applying these methods in material science for a long time, but maybe they just haven't been as widespread. And the thing that's different now is that we have a lot more compute, we have a lot more data that comes from our instruments, and we have the capability now to actually automate much of our data capture. And so this was not possible before, but it's possible now. And that really makes a huge difference when we want to combine machine learning with material science. One example of that is the so-called Emerald Cloud Labs. And so you see a photo here from a Nature article. Emerald Cloud Labs are a synthesis facility based in San Francisco, where if you want to synthesize a particular kind of molecule, you basically provide a recipe, and they go ahead and upload it to this particular cloud lab, which automatically will create that particular molecule based on the synthesis procedures that you tasked it with, right? So in this case, this is although there's no machine learning in this aspect, there is a fully automated chemical synthesis lab that is capable of reproducible science based on the kinds of input that you provided in terms of, for example, synthesis recipes. And so this is where I think a lot of user facilities are headed towards in the future. Can we automate much of these kinds of processes, A, so that they become a lot more efficient, but B, also so they can become a lot more reproducible, right? So how many times have we found a study that we said, okay, if only I had access to that material, I think I could do so many different things. Well, if we had these kinds of labs everywhere, then what we could do is we could find a recipe that was published with a paper. Then we could say, okay, I want this particular material to be synthesized for me. Can you go ahead and do it? And it'll be uploaded to a system, and then that material could be shipped out to you. So this is something where I think the future is going with respect to material synthesis and reproducible and automated science. Just so we get some definitions clear, there is a difference between an automated system and an autonomous system. And I'm going to explain that difference to you with this slide. So an automated system is one where, for example, let's say that you have to water the lawn in the morning at 6 a.m. And you have a timer. And what it does is, as soon as the time hits 6 a.m., this sprinkler turns on, and then after half an hour it turns off, right? So this is an automated system, right? It's capable of turning itself on and off but it doesn't change, right? So a human has to go there to change the times, but otherwise it's going to turn on and turn off at the same time every single day. And there's no real intelligence into the system. Now, if we inject a decision-making capability into the system, an autonomous decision-making capability, then we can convert it from an automated system to an autonomous system. So imagine here that instead of having just the automated sprinkler system with a timer, we also have, for example, a moisture meter, which determines whether the grass is already wet, for example, maybe because it was raining overnight. Well, in this case, what we can do is we can have a decision that's made based on that sensor reading, and based on that, it can decide whether it wants to turn on and for how long it wants to turn on. So this is where there's a difference between a fully automated system and actually a fully autonomous system. And in the autonomous system, you have the automation, but you also have a decision-making capability that's layered on top of that. And usually that decision-making capability implies that you should have some kind of sensor data that goes into the system. Okay. So now that we have that, what we are focused on is autonomous laboratories that are capable of this kind of decision-making and physics discovery. Now, the first thing to notice is that we're not doing this purely for efficiency gains, right? It's not that, for example, we just want to improve our efficiency of our microscopy by 20, 30%. Now, that's actually not going to be a huge win for us, because what that means is that although you might save a little bit of time, in practice, the bottlenecks in science don't tend to be there, right? We need to find out where the bottlenecks are in science, and usually it's not, for example, in how long it takes to characterize a material system. On the other hand, if we have an automated and autonomous system, we can perform new kinds of experiments that previously we could not perform. And so that's really where the big gain is, in my opinion. If we can perform new kinds of experiments and new kinds of characterization and synthesis modalities that we couldn't do under a human-operated paradigm, then I think then we have a big win by moving towards these kinds of systems. And so I'm going to explain to you in this talk a couple of different examples where I'll show you experiments that you could not perform using traditional human-operated paradigm, right? And so those are qualitatively different. It's not a quantitative difference, it's a qualitative difference in terms of the kinds of science that we are capable of doing with this new paradigm. So the first half of my talk, I'm going to discuss autonomous microscopy and some of our efforts there. I am a microscopist by training. I've been studying with atomic force microscopy for more than 15 years. And indeed, the experience in our group goes back more than 20 years. And on the right-hand side, I will also discuss autonomous synthesis methods as well. After we discuss autonomous microscopy. So first, let's talk about how we automate our microscopy. We have a package called Acroscopy, which is automated experiments for microscopy. And basically what happens is that when users want to come to our center and perform an experiment, they can download this package and they can write the experiment that they want to perform in Python code. It's very straightforward, it's very simple. There are some functions that they can call, and then based on those functions, we can run them in a Jupyter notebook. We can upload them to the microscope. We can run on multiple different microscope platforms. It's not limited to just one vendor. We have 4 or 5 different vendors that we can support. And so this runs on some hardware that we custom designed. It gets converted into signals that go to the microscope, and then the microscope will then perform that experiment and spit out some results in an open source file format, which is described in this particular paper here. I won't discuss that, but suffice to say that this is the way that we would like to do science now. Rather than have the human have to be sitting there and control all the parameters themselves by a mouse and keyboard, it would be preferable if we could write some code, have the microscope make decisions, and then spit out those results for the human so that we can actually spend time on the science, that is things like design of experiments, analysis of data, and so forth. So how can we apply this particular automated system to a particular science problem? I am a ferroelectrics person by training, and so I'm very interested in understanding how ferroelectric materials work. So ferroelectric materials have a spontaneous electric polarization. They have they exhibit electrical hysteresis. So when you cycle a ferroelectric with an electric field, you can switch between different polarization states. And they also have regions of aligned polarization called domains, which are separated by domain walls. And so you have these very nice, pretty pictures of ferroelectrics, which you can capture by piezo response force microscopy. And so you can see these domains and these domain walls in these images. And the interesting thing about this is that the motion of these domain walls in ferroelectric materials can govern maybe 30 to 50% of the dielectric and piezoelectric properties of that particular ferroelectric ceramic. And so as a result, we really want to understand how these domain walls in these systems move, how they're pinned by different defects, and how they respond to different electrical and elastic perturbations. So we can do this with our microscope. We can probe where the domain walls are, so we can image them like this, and we can also apply different stimuli to them. We can apply pressure, we can apply electrical voltage through a scanning probe tip, and then we can force them to move, and we can image that as that process is happening. So let's give you a very simple example. Let's imagine that we have a domain wall which we write into the sample. You can see that in the center of the image there. So this is written by an AFM tip that's basically scanning with a voltage across it. And so we can write these domain walls by simply applying voltage to that AFM tip while we're scanning. And what we're interested in is we're interested in understanding how the local domain structure impacts the domain wall pinning. So you can see, for example, that this particular domain wall actually cuts through a bunch of different needle-like domain structures, and we are interested in seeing how this wall will react to different electrical voltage amplitudes and pulse widths. And so what we do is we set up an automated experiment. So we can poll this domain wall in the center of the image. We can image a single line, right? That's that green line there in that image. We can locate the position of our written domain wall, which is basically in the center, and then we can move the tip to that center. We can apply voltage, and then we can image that line again. And we can repeat this process for different values of that voltage. And so the goal here would be to understand how every segment of that domain wall is responding to our applied electrical voltage. So this is work that was performed by one of our students, Mani Valetti, who was at University of Tennessee in Knoxville, but who's now a postdoc at Brookhaven. So this data actually takes about 2.5 days to acquire. So this is fully automated, and so this is a kind of a video that was made after all of that data was acquired. And so again, being a fully automated system, you couldn't do this if you had to do this manually. But we can perform this experiment, and we just set it up on a Friday evening, come back Monday morning, and our experiment is done. And so that's great. And so you can see here that as a result of applying these voltages on every line, the domain wall has been displaced. And so you can also see that the length to which the domain wall actually moves is dependent on its surrounding domain structure, right? It can get pinned by different elastic interfaces. In some cases, it's attracted to these interfaces, and in other cases, it's repelled based on the geometry that we're seeing. So let's have a look at this data set in more detail. Let's take a particular line of data from here. So each line is actually consists of about 180 seconds of scanning with different voltages that are being applied to it. And so if we look here in this particular example, you can see that as a function of time, what happens is that a voltage is being applied to the domain wall every 5 lines or so, and the voltage amplitude that we apply is increasing. And so what happens is that at a particular threshold voltage, the wall starts to displace. And so you can see that there in the white boxed region, that the domain wall that we highlight in blue has actually shifted a little bit from its original position, right? And so if we plot this more neatly, you can see that indeed there's a particular voltage at which we can depin this domain wall, and so this is a depinning voltage, and we can capture this depinning voltage for every single position along that domain wall in that image. But there's another thing that we can see here, which is really interesting, is that after we apply a certain voltage beyond the depinning voltage, which is shown by those red dots, you can see that the domain wall actually extrudes outwards before it relaxes back a little bit. So this sort of snapback effect that we are seeing here is really probably due to the fact that there's an elastic energy penalty from the domain wall bulging out forward and then sort of relaxing back to a lower energy configuration. And so we can actually see that through this automated experiment. We would not be able to do this experiment, obviously, if it was not automated. But this is a very, very straightforward automated experiment. Can we do something more interesting with this kind of microscope? And the answer is absolutely yes. So one of the key features of microscopy is that we can actually correlate the local microstructural features of our sample with the local functional properties. So what I mean by that is you can image local patches of your sample, and then we can capture spectra at those particular patches, and we can find a mapping between the local structure and the local functional property, which is represented by that particular spectra. Now, the problem with this in most microscopy characterization techniques is that this is very time consuming, right? And also, for example, your sample may not last that long. For example, if you're trying to do this in an electron microscope under, let's say, an EEL spectroscopy and so forth. So we want to be smarter about the way that we actually do this kind of characterization. Instead of capturing the spectrum at every individual position along our sample, we can just measure a small batch of points. Maybe we measure 5 or 10% of pixels, but we can then use machine learning methods to be able to inform the microscope as to what is the best set of conditions, or in other words, what is the best location to measure to optimize some particular target metric. And so we do this using GPU servers, which are connected to our instruments. And so with some data pre-processing and some Bayesian optimization approaches, we can tell the microscope where to move next in a fully online way, so that we can adaptively sample our system to understand these structure-property relationships in real time. So in order to do this, we leverage an approach called deep kernel learning, which was proposed by Andrew Gordon Wilson in 2015. So this is a kind of machine learning where we can combine the flexibility of deep neural networks, so-called deep kernels, with the uncertainty quantification from a Gaussian process regression layer. I won't go into the details here. It's not so necessary here. But suffice to say that with this particular kind of machine learning model, we can start to automatically map that structure to function relationship that I was mentioning before. We can get these image patches, which come from our imaging system, and then we can collect spectra at particular individual positions, for example, in this case, hysteresis loops. And then we can apply some kind of scalarizer function to this hysteresis loop. So maybe we want to look at the area of these hysteresis loops. So we can correlate the local image or the local domain structure to the area of the hysteresis loop, and then use this to drive an optimization algorithm that tells the microscope where to sample so that we can optimize for the area of this particular hysteresis loop, right? So that's one example. This is how it works in practice. So you can see here a red dot on the bottom right-hand side, which is moving to different positions on the sample. It is collecting spectra. Those spectra are then getting pushed to a GPU server, which is doing some calculations, and then telling the microscope where to go next. So this is all real time, and it's happening in real time. And so this is a way that we can acquire functional property information with maybe 95% of the veracity of a ground truth experiment, but with only 10% of the time, realistically. So rather than having to sample the entire space, we can sample only 10% of the pixels and acquire, basically the same amount of information using these kinds of methods. We don't have to do this only on the scanning probe microscope. We can also do this on the electron microscope. So this is work by Kevin Roca-Priori, where he's looking at bulk and edge plasmons in MnSP3. And so he's looking to try to find where the edge plasmon is the strongest. And so it turns out that when you try to optimize for this, the algorithm natively seems to prefer particular edges of this structure. We're not exactly sure why, but it certainly seems to be correct. And so there's some real physics that's being learned in this particular process when we try and do this optimization. And we couldn't do this optimization, by the way. We couldn't do this experiment in a traditional mode, because if you expose the sample to too much beam damage, to too much of the electron beam dose, then it does get damaged over time. And so these are very important, in particular, for electron microscopy studies. But there's a couple of issues here. The first issue is that the simple Bayesian optimization is typically low dimensional. We are only looking at low dimensional parameter spaces. And so we can't necessarily look at the entire spectra, right? We need to find a scalarizer function that is going to reduce our high-dimensional spectra into some low-dimensional space that we can then optimize. And so this is one of the issues with traditional Bayesian optimization, is that we need that scalarizer function, and we need it to be known before our experiment starts. We need to know what we're optimizing before we start. But if you actually talk to most microscopists, like myself, we actually don't often know what we're looking for before we start to see it. And so this is a problem. So how do we solve that? I'll explain one way that we have solved that. Another issue here is that even if we don't know the scalarizer, we may not even be able to know what it is that we're after other than a good model. So what I mean by this is, The purpose of any kind of microscopy experiment is to find those structure-property relationships, right? That's the purpose. So can we optimize purely for that particular purpose? Is there a way to do that? Is there a way to say, I want knowledge gain, right? Is there a way to encode that in some kind of algorithm? And it turns out that there are ways to do that, and so I'll show you one way that we've done that as well. The first thing to note here is that when we talk about autonomous systems, we don't necessarily mean that we want to take the humans out of the loop. We sometimes want the humans in the loop. And so why is that important? So there's a really nice paper that was published last year about human-machine collaboration for improving semiconductor process development. This is in Nature. And so what it shows is that human AI systems work better together than either system alone, right? So you can have a human expert and they will come to some optimization problem and they will solve it. Or you can have an AI system which tries to solve that optimization problem. But it turns out that if you have the right mix of human and AI agents, then they can solve the problem better than if you were trying to use each of those alone. And so the reason is because AI systems typically don't have prior knowledge about the domain, whereas human experts actually do. And so what you want to do is you want to inject the right amount of human domain knowledge into AI algorithms, either by having a human in the loop or by injecting that information somehow mathematically. And so it's really important to know that just because we say that we're creating autonomous laboratories, that doesn't mean that we're taking the human out of the lab, right? The human expert is still absolutely required and indeed advantageous for many of these operations. Okay. So how do we do that here? So one way that we've done that here is that we have created what's called the Bayesian optimization active recommender system. So this is work done by Arpan Biswas, who is a postdoc of ours, who recently became a research professor at the University of Tennessee Oak Ridge Innovation Institute. And so the idea here is that when you are on a microscope and collecting spectra, you can rate these spectra. Maybe you don't know what's good about them or what's great, what's a scalarizer function. In other words, you don't know whether you are looking for a particular feature or not, but you can rate them as to whether they're good or bad, and you'd like to see more of that kind of spectra, right? So this is very similar to a kind of social media feed. Do you like these posts? Do you not like these posts? Based on what you see, the microscope will be calibrated to try to find more of what you like and less of what you don't like. Right? And so this is a kind of recommender system that we can utilize to have that human expert in the loop, but also have the AI optimized so that they are aligned for the same objective. And so this is an example. Imagine that you have a particular spectra that's being captured by that red X there on the left-hand side image. On the right-hand side, what happens is that after you rate a number of spectra that you capture, there's a target function that's being acquired. In other words, there's a weighted sum of the spectra that you have upvoted. And this is the target function that will be tried to be optimized for. In other words, the microscope will try to find spectra that look closer to your target function. And so this is it. This is the example running in real time. You can see our staff scientist, Yongcha Lu, he's on the microscope, he's collecting some spectra, and he is being asked to vote on the spectra as to whether this is a good spectra, whether it's a very good spectra, or whether it's a bad spectra. And based on those voting patterns, the system will automatically determine where is the place to sample next, so that we can align the sampling to what the user actually wants. And you might think, oh, this is, relatively like a novelty, but it's not particularly important. It turns out it's actually very important because a lot of the time we cannot express what we want in terms of a mathematical formulation as a user or as an experimentalist. And so this really becomes handy when you cannot express the features of the spectra that are necessary for you to be interested in. And so this is one way that we can overcome that particular block. And indeed, these kinds of human AI collaborations are going to be a lot more important when we move towards these autonomous systems. The other thing we can do here is we can use what's called curiosity-based optimization. So what is the goal of science? And the goal of science is, in particular, is to advance our understanding based on what we can predict for different conditions, right? If we can predict the universe under all different kinds of conditions, we know what's going to happen next, and therefore we can adjust accordingly. Now, the way that that usually works is that simulations or modelers or theoreticians predict for different kinds of, for example, spatial ranges, energy ranges, and so forth. And experimentalists try to break those predictions, right? So whenever there's a break in that prediction, and whenever an experimentalist has found something that's incompatible with an existing model, well, that's where the new physics actually lies, right? And so what we want to do is we want to apply that same notion of what's called curiosity. In this case, curiosity is being defined as a deviation from what we expected. We expected something, we have a model of the world, and we expect something to happen. When this doesn't happen, then we should reward that, right? We should try to find points in the parameter space where our model is struggling. And that's the purpose. If we can do that, then we can acquire more data in those regions, and hopefully there's new physics to be found in those particular areas. And so the way that we can do that is we can have what's called an error predictor model. So this is work that was done by a student, Rama Vasudevan, who was at University of North Carolina. And he stayed with us over the summer, and he created this algorithm where effectively we have a simple neural network which takes our images and predicts the local spectral properties from those image patches. But we have another model which is going to predict the error in that particular model, right? And so by having another model which is going to predict error, we can predict where the error is going to be largest and try to sample those particular regions. And so we call this sort of curiosity-based optimization. And just so that you know that it works, it actually, if we apply it for example, a lead-tight net thin film, and we acquire maybe 100 different spectra from it using this particular method, you can see that the measured value of the spectra and the predicted spectra are actually quite close, so it's doing a pretty good job. But you can also see that if we run this algorithm multiple times to gauge its efficiency, on the left-hand side here, you can see the curiosity-based optimization has a lower loss compared to a random search method. So what this is telling us is that by using this particular approach, we're able to more quickly create data-driven models that minimize their own error, right? And so that's the whole purpose here. Can we create models that will minimize their error, or maybe find where that error is not reducing, and so that we can inform us as to how to improve those particular models. We can also do manipulation with our microscope. It's not limited to simple characterization, right? And so we can start to use autonomous workflows to manipulate our materials, either with the scanning probe, or with the electron beam, or with the scanning tunneling microscope chip. We can do all of those three as well. I won't focus on the electron microscopy today part, but you can read about it in a particular paper by Kevin Rocco-Prioria, where he's looking at how to create different kinds of defect structures in graphene using a combination of AI, machine learning, and automation. I will talk to you about how we can modify domain walls, though. And so imagine that what we want to do is we want to be able to modify these domain walls in this lead-tight net thin film. We can apply different voltages at different pulse widths and different amplitudes, and then we can image them and see what happens afterwards, right? Well, what we can do is we can create a surrogate model, which is going to acquire, first we're going to acquire some data, and we can acquire the data using the automated microscope. So what's happening here is that the tip is moving to different positions along the domain wall, applying voltages, and then imaging what's happening. And it's doing this in a fully autonomous fashion. And over a couple of days, you can collect data that looks like this, where every particular point is a different sort of what we call state transition, where the tip is moving to that point. It has a particular action, in other words, a different voltage, amplitude, and pulse width, and then we can measure what happens to the domain wall as a result of that particular action. And so this is, again, a fully automated system. But now what we have is we have hundreds of these state transitions that we can put into a neural network model. And so this neural network model takes as input the existing state, the existing kind of shape of the domain wall, as well as the action that we perform. What is the voltage that we're applying? Where are we applying it? You know, how long does it go? How long does it is it applied for? And what is the amplitude of this excitation? So we put these as input parameters to our neural network, and then as an output, we want to know what's going to happen to the domain wall as a result of applying this kind of sequence of pulses, right? So this is our surrogate model. There's some physics regularization in here, but it's basically data-driven. And so being a data-driven model, we can also very quickly investigate what happens when we change input parameters. So now once the model is trained, what we're doing here is we are looking at how the domain wall is responding for different values of the voltage and different values of the pulse width, as well as for different initial starting conditions. We can say, for example, when the domain wall is flat and we apply a certain voltage, it's going to respond a certain way. But when the domain wall is already bulged, when we apply the same voltage, it's going to respond in a very different way. And the reason for this is obviously because the way that the domain wall moves is obviously related to things like the elastic energy penalties and electrical energy penalties that are associated with that configuration. And as those configurations change, we expect the dynamics to change. And we actually encode those dynamics, or we have been encoding those dynamics in these learned surrogate models. We can then use these surrogate models with reinforcement learning agents to tell us what kind of sequence of pulses we should apply to the domain wall to achieve a desired configuration. So on the right-hand side, you see a dashed red line. This is the desired domain wall configuration that we want. And then this is using the surrogate model. A reinforcement learning agent has determined what is the sequence of pulses that should be applied. And so you can see that the mean absolute error here is pretty small between what the result is and what the domain wall profile is that we would like to obtain. This is just like a demonstration. It's not actually this is purely done with the surrogate model, and the next step is to apply it on the microscope. But it shows the potential of these data-driven models for us to be able to use for materials manipulation in an autonomous fashion. So I'm going to focus next on autonomous synthesis. And so this is some work that was largely done by Sumner Harris. So he basically built an autonomous pulse laser deposition platform. And so in pulse laser deposition, this is a way that we can grow thin films, which probably most of you are very familiar with. There's an excimer laser, which strikes a target of the material that you're trying to grow. It forms a plasma, which then condenses onto a substrate. The substrate is heated. And so we can control the pulses of the laser. We can control the energy of those pulses. We can control background gas pressures, and we can control the temperature of the substrate. And based on all of these different parameters, we can grow thin films of very different kinds of materials and with very interesting kinds of properties. Now, traditionally, this is done all manually, right? A graduate student or a postdoc would be in the lab and toiling away for months at a time to optimize parameters for a thin film deposition. In this particular case, in this system, Sumner built a fully automated PLD platform where we can effectively upload a set of synthesis conditions, and the synthesis chamber will go ahead and try those conditions, synthesize that film, characterize it, and then give the results back to the user. Right? So it can really short-circuit that development time from months to days. So we did this for tungsten diselenide. You can grow 2D materials as well using pulse laser deposition. And so what happens here is that there's a 4-dimensional parameter space to optimize: the pressure of the background gas, the temperature of the substrate, and the energy of the lasers. And so we can navigate this space using the same algorithms that we use to navigate the space in the microscopy setting, right? And so this was used as a general workflow where what happens is that after the film is deposited, there's an in situ Raman measurement. Again, this is also fully automated. And based on the Raman measurement, we can score whether the film is of high quality or not. And then we can optimize for that film quality based on this Raman score. And so when you try this in practice, you find that actually the model that is learned in the process of this autonomous exploration actually finds 2 different growth regimes for these kinds of films, which is very interesting. There's a low kinetic energy growth regime, and there's a slightly higher kinetic energy growth regime, and this is found autonomously by this particular algorithm. The other thing that you can note is that if you look at the graph on the top right there, you can see that there's a particular film that's grown after about 80 samples, which has a very high Raman score, and this is higher than what our experimentalists could produce, our expert experimentalists could produce. So the AI is certainly capable of optimizing in a fashion that's superior to the human expert. Now, you also notice that there's a lot of failed samples there where the Raman score is basically 0, and so that's basically because the system is trying to explore the parameter space, and it's doing so in a very naive fashion. So the question is, can we make it less naive by having a human or having some other system say that, okay, the Raman score in these areas looks like it's very poor, you should try to avoid those areas. And so that's exactly what Yongtao Liu did. He created a method called a dual Gaussian process method, where instead of having a single optimization algorithm, which is only optimizing for that Raman score. There's a second optimization that's happening for the quality of the spectra that are actually being acquired. So if the spectra are of poor quality, then what's happening is that there's a gate that effectively says you should stop exploration in these spaces where the spectra are of really poor quality. And by doing this, we can actually reduce those wasted samples by a very significant fraction. And so this is work that's very recent, and I believe it's on archive as of last week. We can go further than this. We don't have to necessarily wait for the film to be completed before we understand how it's actually proceeding. So one thing that we have access to in this chamber is in situ imaging of the plume as the film is being grown. And so what you see on the top left there is an ICCD image sequence of the plume as it's interacting with the sample. And so what happens over time as a function of each of these pulses. And so we have all these images, and we can start to create deep learning models or machine learning models that correlate the image sequence that we can capture during the deposition with kinetic growth models or parameters of kinetic growth models for our particular film deposition. This is really powerful because what it suggests is that merely by observing the process of that deposition using these CCDs, we can start to control the growth in principle because we can start to understand the kinetics of that particular growth process, right? So this is the first step in realizing real-time control over our synthesis. It's that we can start to use data-driven models for this real-time parameter estimation in this way. But if we really want to have strong control over synthesis, what we need is what's called so-called state estimation or parameter estimation. We should have a model for how our film is growing, and we should be able to infer the parameters of that model as a function of the deposition so that we can change it over time, right? So the real goal in material synthesis is to, for example, achieve some desired metastable state or some desired monon-equilibrium phase. So how are we going to do that? Well, the real way we're going to do that is if we have control over our synthesis during the synthesis process. It's not enough just to say, okay, I'm going to have a set of conditions, run my experiment, and come back, right? There's a lot of processing that can happen in between that rely on kinetic effects that we really need to be taking care of, and we really need to understand and control. And so we can actually use what's called parameter estimation or state estimation methods from model-based control methods to be able to do this for us. This is used, for example, in self-driving cars, but we need to use it for synthesis as well. And the method that we're using is something called particle filtering, and this is something that is being developed by Professor Feng Bao at the University of Florida, and so he's a real pioneer in these kinds of efforts, and so we've been working with him and his team to implement some of the mathematical advances that they're creating onto our pulse laser deposition platforms. So let's talk about a very simple two-step kinetic model for a growth of a thin film. Imagine that you have a nucleation process. As your plasma condenses onto the sample, you have some growth, you have some nucleation of that layer, and then you have some growth of that layer. And so there are two effective parameters. There's a nucleation coefficient, and then there's a growth coefficient. And we can measure our film as it's growing based on reflectivity measurements. So we have a real-time observation of our film as it's growing based on the Fresnel equations. We can calculate what we should expect for some layer coverage. And if we actually do this for this very simple model, you can see that as we grow different layers, our contrast starts to drop in terms of our reflectivity. This is obvious. This is quite straightforward. But the point is that can we predict how this contrast is going to be with only a few seconds of data that we capture on the device, right? That's the goal. Because if we start to predict how this contrast is going to behave, then we can start to change the way that we're depositing to achieve some desired, let's say, layer coverage. So does this work on simulated data? This particle filtering method, actually, we tried it on some simulated data. And so what you see here is on the left-hand side, you see the nucleation. In the middle, you see the growth parameter. And as a function of time, what the system is estimating for these parameters. The dashed black line are the actual ground truths. And you can see that after about 20 seconds, the particle filtering method does seem to converge on what is the actual growth coefficient and what is the actual nucleation coefficient. And we can predict exactly what the contrast is going to look like. That's that green line on the right-hand side. You can clearly see that, at least for the synthetic data, it is perfectly possible for us to use these methods in real time to be able to understand the parameters of a particular growth model. And so this is it applied to real data. This is still in an offline testing mode, but it is actually real data. And so what we can do is, again, it's the same kind of nucleation and growth model. And what we're trying to do is seeing if it converges to a particular value for that nucleation and growth, and seeing how the predictions diverge over time. So on the bottom right-hand side, you can see the prediction time at 8 seconds, 11 seconds, 15, and 16 seconds. Obviously, the longer you wait, the smaller the uncertainty in your predictions. But even at 8 seconds, you have a pretty good idea of where the film is going to be after 30 seconds. So in other words, you can predict 20 seconds into the future using this kind of model, and it's a very simple model. We can augment this model with more complicated parameters, and we can probably get better results than this. But the point is that this allows us, for the first time, to really do model-based control in PLD, which has never been done before, and I think is going to be critical if we're going to realize PLD in terms of being able to reproducibly create non-equilibrium and metastable states with desired functional properties. So I want to end now with some future directions. I've shown you some of the advances that we've made in characterization and some advances that we've made in synthesis in terms of our autonomous capabilities. But I also think that there's been a lot going on in terms of just the larger autonomous landscape in society, as well as the emergence of things like foundation models. And I thought it would be useful to discuss where those could be, how those fit in with this particular understanding of where science is heading in the so-called smart labs. So the first thing is that you've all probably used ChatGPT before. You've probably used other kinds of foundation models. And so large language models are ubiquitous these days. They have their issues, of course, but I think that when we can use them for what they're designed for, in other words, they are very good at distilling large volumes of literature and then summarizing them for us. And so if we can use them for that particular purpose, I think they can be really great AI assistants when we are there in the lab trying to design experiments. The other thing that they're really good for is code generation. So I mentioned before that users come to our center, they write code, they deploy it on the instrument, and then the instrument performs the experiment. Is it really necessary to write that code and learn Python? Well, maybe not. We've actually have a paper in MLSC that should be coming out soon. It's been accepted, where we basically drop our package into ChatGPT and then converse with it in natural language and say, I want this kind of experiment to be performed. Can you generate the code for me that's going to do this? It turns out it works pretty well for simple kinds of experiments. And so it can be a good assistant to be able to interface with these autonomous systems. The other thing that it can be really helpful for is workflow planning and execution, right? So based on some data that is being collected, maybe we want to organize a simulation run, for instance. And so LLMs can be quite handy at being sort of the execution and workflow manager in these kinds of cases, to be able to look at data from multiple different sources, summarize it, and then provide it for the user for the purpose of workflow execution. The issues with LLMs right now are that, of course, there's hallucination, but the other problem is that most of these LLMs are not fine-tuned for scientific data, and so although they're kind of very strong for things like chemical synthesis, because they have some kind of understanding of, or there's some kind of inbuilt training data for chemistry, for the things that are really important for us, like understanding how disordered materials work. Right now, LLMs really lack that kind of context. And so we really need scientific LLMs, as it were, LLMs that are trained on scientific data so that we can really turbocharge their use within these particular applications. The other aspect here that's very important is multi-step workflow optimization. So with Sergey and with some of my colleagues, we wrote a paper in Applied Physics Reviews where we talk about the fact that in science, you're never just performing one experiment. You have a synthesis, you have a characterization, you have synthesis. It's really a workflow, right? And so the question is, based on what has already been executed to date on that particular system, what is the best next set of conditions, or what is the best next experiment to perform? What else should you be looking at, right? And so this is a problem which hasn't had a lot of attention in the scientific field, but I think it's going to be more and more important as we move towards smart labs. And if you are interested in that particular aspect, I strongly encourage you to read a multi-step workflow optimization paper that we wrote this year. And finally, if we really are to realize the promise of autonomous laboratories, I don't think we're going to do so by purely data-driven models, as I've shown before. What we really need is we need theory in the loop, right? We need simulations that we can rely on during the experiment and that we can modify during the experiment. And so what we want is we want the navigation of the theoretical space and the navigation of the experimental space to happen at the same time and inform each other. And so this cross kind of co-navigation of both experimental and simulation spaces is going to be the way that we can really close that loop between simulations and experiment. Right now, it takes months or years. We want to basically condense that into the same experimental workflow, right? And so that way we can have much more predictive models that are refined based on data, but at the same time have our data being captured in a way that's beneficial to improving those particular models as well. And so I think that's where we're headed in the future. Finally, if you're interested in anything that I mentioned today, I do have to point out that we are a user facility. So if you'd like to work with me on machine learning or any kind of autonomous experiments and have access to these capabilities, we are a user facility. And if you write a 2-page user proposal, which we're very happy to help you with, you can get free access to our facility and free access to our staff time. And so please come and talk to me if you're interested in collaborations. It's a great venue and a great place to do science. Thank you so much for listening.
Thank you very much, Rama, for this beautiful talk. Questions?
Very nice talk, Rama. I'm wondering how smart is the system in providing feedback in case any changes. For example, if you scan the surface with FM, leave it over the weekend, well, probably when the tip breaks, the system will stop. But a tip may wear off and start giving you low resolution image or artifacts.
Yeah.
Do you get a feedback? Can it be incorporated? Or another, if the system finds something unusual, you may have a discovery made, but it's necessary to alert user about it. Is it possible to do?
Yeah, so the question about the artifacts, or sorry, the question about the system determining whether something is starting to go wrong, that's a really good point, because traditionally that's not something that people often look at. They just automate it and assume that the system is going to remain the same. But in reality, you need metrics to monitor during the experiment, so you can gauge whether something is deviating from the traditional or what you've already captured to date. So I think that's something that we are working on, and it's particularly important for things like obviously for AFM when the tip changes, even electron microscopy when when things go out of focus, you need to come back and align them again. And so we are working on methods to incorporate into these workflows so that happens. So absolutely, that's a problem, and there are methods that need to be developed or are being developed to counter that. As far as when something new is discovered, I think there are ways that we can quantify this. So when you quantify, like Bayesian surprise is one particular kind of way to do that, is that how much does this deviate from our expectation? And so we can try to encourage that kind of exploration. But that can absolutely be used to alert a user to say, like, is this a real change, or is it just an artifact? Because oftentimes that's what happens, is that, oh, you just found an artifact that we've known about for forever. And so these systems are not smart in that sense. They don't know the difference between something new and a new artifact that has just been, because of a double tip or what have you. And so it's really critical to have the human in the loop for all of these operations. I don't see the human is going to be taken away from these operations. What would happen is that we need to develop metrics and visualizations for the user to gauge when things are going wrong and when things are deviating from where they should be. So that AI alignment problem is something that is going to have to be tackled. Absolutely.
Yeah, thank you, Dr. Vasudevan. Very nice talk. So my question is, for example, when we go for large-scale applications like photovoltaics, so we have to do mapping at different places because working with nanosystem is quite complex. And if we have defects, for example, from one place to other, how intelligent is the system to detect, like, we have some different defects in different zones, and it can propose some new steps for the next preparation of samples? How it can work in that way? Thank you.
Yeah, so the goal of the deep kernel learning is really to do exactly what you mentioned, which is to try to find the relationship between the local structural properties or the local structure, maybe the presence of defects, and then the local functional properties. So we have found that it is very capable as long as the information is present in the image in some way, right? So if, for instance, the functional property that's being measured, let's say you measure conductivity, right? Some kind of conductance is being measured, but there is no relationship between the images that you are capturing and the actual local conductance, then we won't be able to learn any correlation, right? But usually that's not the case. Usually there are some kind of features that are present in the image that will correlate. So in that case, yes, absolutely, we've found that you can more or less find and enumerate the defects that are present. So you won't know what defects they are, but you'll be able to know that there are this many types based on the functional responses. So that can be done. So absolutely.
I understand it's very positive. Well, I am a university professor and I have graduate students. As a university professor, I actually do not rely anymore on any report of the lab because they are intelligence. Then I don't, and then I have an exam, and then I say yes, okay, I can. I feel as a prison, as a guard of a prison, trying to torture people to read a book and not to use the ChatGPT to make the Python program. And then you see, you need the human being to feedback. Then I am worried in 3 years we do not have a human to feedback your system. I work with a complex analytical techniques, and I say that a lot, but a huge percentage of the errors in analytical techniques is not misconduct, but overinterpretation. Then I am worried that in your automatic intelligence, soon your natural intelligence feedback will be gone.
Yeah, so I would say that's a fair point and it is something that I wonder about, which is, for example, me being trained traditionally, you get an intuition for a chemical parameter space when you're trying to grow a thin film, right? And if it's fully automated, do you still get that? Or is that something that will be provided to you through some other means? Like, for example, by investigating the surrogate model's plots, will you start to get a different kind of intuition, but it's still important? Ultimately, science is not, the AI is not going to do the interpretation for you, right? Whatever spectra are found, it's only capable of finding those correlations. And whether that's an artifact or whether that's something real, that is going to still be up to the scientists, and we don't envision that that's going to change anytime soon. Yeah.
Thank you for the very nice talk. I am wondering, because we saw very nice solid state examples,
yes.
But there are in physics there are many complex problems that we don't have predictive laws to see this emergent problem, complex systems. Do you have some work on that direction?
So I'm in condensed matter, so traditionally, we do distorted materials. However, certainly in situations where your traditional theory breaks down and it's not capable of describing certain systems, I think that you can definitely rely on, or not rely on, but certainly try data-driven modeling that's present. Maybe you can inform it with some theory, so some broad priors that come from theory for different conditions, but at the same time rely more on the experiment. So ultimately, our ability to describe a system is governed by the quality of that modeling, and the modeling can be fully first principles, or it can be fully data-driven, or somewhere in between. And so for different systems, it's going to fall in a different kind of area on this spectrum. So maybe in the cases where you have absolutely no way of understanding the system using traditional means, you'd use data-driven modeling. An example of that is that ICCD plume imaging. So we can't actually model that. That's actually outside of the realms of possibility for modeling at this stage. However, the data-driven model works very well. We actually don't know exactly how it's working, but the data-driven model is perfectly capable of looking at these plume image sequences and correlating it to properties of the film that we're growing.
I had a question actually on this like, don't like. To me, it's a bit dangerous. I mean, we teach our students not to discard data just because they don't like them.
Yeah.
You need to have a representation of the whole sample. So
Sure.
Isn't there a risk to use this type of algorithm just to look for location that we like and not have a...
Yeah, so we did, in that paper, we actually looked at, so what would happen if you had like a malicious actor or you had random votes, those sorts of things. It's certainly like the case that most of these systems rely on, the operator to be ethical and, to be a domain expert. So the goal here is not that you just have any student that doesn't know anything about the system to be able to say, oh, they like this and don't like this, but rather to have an expert to say that, oh, this is expected, so I'm not particularly interested in this, but there's an interesting feature, the step-like feature that I'm seeing here, and I'd like to see where that's correlated with on the sample, and so it's useful for those situations. But yes, I mean, anytime that you have you're only measuring a small subset of your sample, you run that risk of it not being representative of the entire sample.
Thank you. Other questions?
Thank you, Rama, for the excellent talk. So I was a bit puzzled when you said that you input the parameters and then your system uses those parameters. But what if there is another parameter that you're not considering? So it wouldn't be biasing already your result by limiting the range of parameters. And then a very other quick question on ethics.
Yes.
So if the whole discovery is done by a machine, who owns the discovery?
Yeah, so I don't think I can answer the ethics one in a reasonable amount of time. Maybe over lunch. However, with the modeling itself and the number of parameters that are required, so we have a good estimate as to whether the model is doing a good job in prediction or not, right? So if the model is doing poorly in prediction, then there are ways for us to be able to say, even on the fly, to able to add sort of data-driven terms. So maybe you have a kinetic model, and then you have another model, which is maybe some kind of neural net, which gets added to it to correct for any errors in your existing model. And then we can go and examine where those corrections are happening. So maybe it's only happening in certain temperature ranges or pressure ranges and so forth. So there are ways that we can absolutely determine whether the model is performing poorly, and therefore needs to be changed. So there are metrics that we can look at.
All right, thank you very much. I think we need to stop here. So thank you very much again for the excellent talk. And
this is a craft from the region here in Mexico. It's handmade, it's unique in the world. So thank you very much for your talk, and I hope that you like it.
Thank you so much. It's a great honor.
Thank you, all of you. That's it, right?